Hello everyone, and very warm welcome to another webinar co-organized by A&T Neuro. My name is Farnoosh Safavi. I'm the head of education at A&T and upstream product manager in eMagin. I'm delighted to be moderating today's session on advancements and challenges in EEG source imaging. Our presenter today is Dr. Martin Seeber, who studied electrical and biomedical engineering at the Graz University of Technology and received his PhD in 2017 from the Laboratory of Brain Computing Interfaces. Afterwards, he joined the Functional Brain Mapping Lab of Christophe Michel at the University of Geneva. He currently is working as postdoctoral researcher, focusing on EEG source imaging-based analysis and research of spontaneous large-scale brain dynamics. After his presentation, A&T Neuro application expert and product management team in Berlin will walk you through EEG acquisition as well as ASA source reconstruction software. There will be a short Q&A session afterwards, and you can send your questions already in the questions chat box. Also, you will have some poll questions in the course of the presentation, which require your active participation to keep the session more interactive. So let's get started. Martin, many thanks for accepting our invitation. We're looking forward.
Thank you. And thank you very much for your kind introduction. As Farnoosh already mentioned, I'm a postdoc here in Geneva, and today I'm going to have the great pleasure to talk about advancements and challenges in EEG source imaging. I'm going to dive right into it. First, I'm going to introduce EEG source imaging methods. And afterwards, I will show two recent research examples where we used EEG source imaging technology for mobile body brain imaging. And we're also going to talk about the feasibility or the detectability of subcortical signals using non-invasive high-density EEG. And I'm closing with the validation or discussion about validating source imaging results. As you might know, that source imaging is only an estimate of cortical activity, so it's important to validate the estimates, right? So first of all, let me talk about EEG in comparison to other non-invasive recording techniques. As most of you might know, EEG has a high temporal resolution, it has great coverage, it is silent, mobile, and affordable, and all that taken together makes it a feasible and I believe great imaging technology if you have multi-channel high-density EEG recorded, and if you can combine these multi-channel EEG signals with recent developments in computational approaches, right? So I think EEG has a bright future because the hardware is improving, plus there are more advanced computational methods that are leading to more advanced analysis as source imaging. So that provides new possibilities. And here I want to just guide you through some applications or some distinctions. So here I show two different recording systems that are low density systems but wearable. So there's the CEG grid or the Dream 2. Those systems are fully wearable, but they're not high density. So these systems you can use rather for applications, right? Or to derive some biomarkers. And there's this intermediate setup that is usually used for clinical research or field research. You can see here a wearable EEG cap, and the amplifier is usually in a backpack or remote, and these systems are usually used in a clinical setting or a field setting. But if you're more interested in basic research, you usually go even more high density, where you use these high density EEG systems. Here I show two examples from EGI, but also the waveguard from A&T. And those you can use for rather basic research to answer your scientific questions. So what I want to state with this slide is, depending on your application or depending on your research question, you could pick the right system, either fully applications, something intermediate like clinical or field research, or basic research. Depending on that, it is wise to choose the right system, right? So we have already arrived at the first question of the Quickpoll. That is, how many EEG channels do you mostly use in your study? Do you use usually below 32, 64, 128, 256, or none?
Okay, the poll is ongoing, and let's give it 30 seconds. Okay, we close the poll and share the results. Interestingly, the majority, 53%, voted for 64 channel, around 64 channel EEG system, then 32 channel or less, then 11%, 128, 2%, 256, and 4% none.
Yeah, that makes sense to me. So that's also the most common number that is usually used. It's perfectly fine. There is no right answer, of course. It depends on your research question, as I said before. So here I want to introduce source imaging or source modeling. First, we have to build a model of how the field spread from the brain to the scalp. And then we want to derive, given this model and the EEG recordings, we want to reconstruct this ground truth patch that I was simulating here from the scalp recordings, right? So we're going to do this using a forward model or a head model, and this head model we use to compute or describe how the brain activity spreads to the scalp, right? When we record EEG, of course, we just have this information, but we want to find out where are the underlying sources, and that we have to do with inverse source imaging. So the second step is to combine all these multi-channel EEG recordings to a topography, and using this topography or multi-channel co-registered data, we can compute inversion of the signals to do source localization. And I'm going to close up, as I said before, with a validation discussion. So let's first tackle the first point here, that is field and head modeling. So in this example, I did a simulation like all the dipoles are pointing up. So in that case, you can see already from the topography, you can more or less guess where the sources were, right? But that's not always the case. That's only the case if you have a radial dipole underlying. So if you have a radial dipole pointing from here to there, there's a positivity here and a negativity there, right? So we have here indicated in red, the positive maximum here, and all the surrounding on the lower part here is all negative, right? So from this topography, you would assume, okay, the source is somewhere here. But that is kind of not the case if you have a tangential dipole. If you would have a tangential dipole, we have a positivity here and negativity here, right? So what we would observe on the topography is a positivity on the front of the head and the negativity on the back of the head, right? So from this topography, you would do the wrong interpretation if you would just look at the peak on the channel space, right? So you already can see that this is somewhat problematic, that the underlying sources, depending how they're orientated or how they're configured, lead to a different topography, and the topography is not necessarily guiding you into the right direction for correct interpretation, right? And for that, source imaging is needed. So there is a single channel analysis. So if you would look just on a single channel, you can look at the whole topography, what is way better already because it gives you a more complete picture. But then the third step is source imaging or source modeling. So in field modeling, as I said, we want to describe how the sources spread to the scalp. And the most important influential factors is the geometry and conductivity values. We want to use different conductivity values related to specific tissues, meaning brain tissue, skull tissue, and scalp tissue, right? And the most classic approach how to do this is using spherical models. So you assume that the head is a sphere or made out of separate sphere with different conductivity values, right? So you can then compute, because it's all spherical, how these sources would spread to the scalp. On the scalp, you can measure a specific voltage, right? That we record with the EEG. So you can do this in this very classical way using spheres. You can use boundary element models where you can move away from the spheres having individual shapes, right? Because the head is not a sphere. So you can include or model more complex surfaces that are not necessarily spherical, but the principle is the same. You always compute from one surface to the next surface, and from that surface to the next surface, and you assume that all the volume have constant, mostly constant conductivity values, and with this you have already the conductivity values for the given volumes, and you compute always from surface to surface until you arrive at the scalp, and at the scalp you know that there are the EEG recordings. So here you can have just a more advanced model than before. And the even more advanced model, the most recent development, are FEM models, where you have a fully individualized or realistic model of your head. You can approximate the surfaces or the volumes of the different tissues. Again, here the brain, the skull, and the scalp with triangles. And there you do the computations, or how the field spread. You can compute that from every point to every other point, from triangle to triangle, right? So all these three approaches have the same aim, because what we want to do is make a relation between the sources and the scalp recordings, right? Here we have these source densities, small dipoles here, as I show here in red arrows, but what we measure is only the EEG, right? So we want to make a relation between those two based on how the electric field spread. And usually how you do this, so these are the three different ways how people do that. It's just you make you make a head model, and this head model provides you this large K matrix, what we call the forward model, right? So that's a matrix that transforms the sources to the scalp recordings, right? So that is also the nice thing, that you can have a linear equation describing you the relation between the sources and the scalp recordings. So this K matrix is of the dimension number of sources times the number of channels, right? So if you would have a specific number of sources here, you multiply that by K, and this K is only dependent on this field model here. And the field model is primarily dependent on the conductivity values and the geometry. In the geometry, I mean how far things are apart. So how distant is source A to electrode B, for instance, right? So that is the whole idea. So we can move that a bit further. No, let's do a question. So in this poll, we're going to ask, on what spatial level do you analyze your EEG data? Do you analyze single channels? Do you analyze topographic data? Do you analyze dipoles? Or do you analyze distributed source estimates?
Okay, let's give this 30 seconds or so. All right, around 66% voted. We close the poll and share the results. Topographic EEG analysis has the highest vote, 47%. Then dipole localization, 21%. Single channel, 16%. Distributed sources, 9%. And 7% selected none.
Okay, great. So we have already some room for source localization here, because it's only 9%. It's all valid that you look into topographies. It's all great. I just of course, very convinced of EEG source imaging, and that's why we discuss it today. So here I show an example of individualized, almost ready-to-go head model or head models. I show here in 4 different cortical surfaces, and as you can see, those come from 4 different individuals, and they are quite different in size, shape, and cortical folding, right? So depending on the individual, those the brain and the head is of course quite different, right? And then you can use this cortical reconstruction to build up a forward model, as I discussed before. And here you can see already the brain is integrated in these skull surfaces and the scalp surfaces, and on top of the scalp surface there are the electrodes, right? So here everything is already nicely in place, and as I said, the geometry makes a big difference or plays a big role. That is the relation between every solution point here or every source point here to every electrode, right? So that is very important. So this is all sorted out. And the assumptions of the conductivity values is of course very, very influential too for head modeling. So assuming this is all done and this is all in place, we can come from the sources, having this model in place and compute in a model given a specific source configuration, a specific scalp configuration, right? So this is very well defined. So based on physics and knowing how the electrical field spread, this is all described since about 100 years. We can make this model. So this is more or less the easy part. The more challenging part is inverting. So if we have a specific topography, if we have all the channels integrated in this model in place, we want to invert this linear relation I was talking before, right? So we don't want to describe how the field spread from the brain to the scalp, but we want to estimate the sources from the EEG recording, right? And that we do in somewhat inverting this forward model, right? So T is some inverse of K, right? But because we usually want to estimate more sources in the brain than we have recorded channels, we have to include additional constraints, right? Here is the very often used in classical minimum norm solution, where usually you say your estimates times K must match your recordings, that is phi, right? So this term should be small, plus the sources themselves should be small, right? That's why it's called the minimum norm solution, because of course the estimate should explain the data, but also the sources themselves should be small, right? So one effect of this is they then usually don't cancel each other, right? And here is the whole family. So this is all possibility of inverse solution families. There are 2 different assumptions in the solution space already. You can go for dipole model, or you can go for distributed models, right? That's already profoundly different. So if you have already a topography that very much looks dipolar, and if you already know in your study you look for only 1 or 2 dipoles, you can go for a dipole model, right? Somehow the disadvantage is that you have to a priori set how many dipoles there are in the brain, right? And if you don't guess this number, or if you don't have this information ready, and if you're not sure how many dipoles are in your study or at the time where you want to look or localize your data, then probably distributed source models would be more appropriate, right? So as I said, there is this classic family of minimum norm solutions that has different variants, and very related to this, there are also these nonlinear post-hoc normalization methods, including S-Loretta, where you also have additional constraints somehow saying we want to have uniform power distribution, right? A very different family is the beamformer. In a beamformer, you try to maximize for every solution point the SNR of that at that point, and minimize all the influences of all the others, right? And that you do for every solution point. And because you do that for every solution point, the estimates not necessarily describe all the data you recorded on the scalp, right? but beamforming is great for source reconstruction, more or less reconstructing the waveforms, not only necessarily localization, right? And then the most recent framework is the sparse and Bayesian framework, where you usually have sparsity constraints, meaning you look for several patches in the brain that should be sharp, right? For instance, here there is this example of the iris, where the constraint is that the edges of your patches should be steep, and everything else should be zero, right? So that's somehow the difference between all of the different families. It really depends a bit on your research question and the application, which one to pick, right? So I think it's worth it to check what's there and what's already implemented, and then choose depending on your research question the appropriate method. And having that said, you don't have to do all that by yourself, right? So there's this huge work done in the last more or less 20 years or 30 years, and this is all implemented in several toolboxes. So I want to suggest these toolboxes. There are different ones depending on how experienced you are in coding, or if you more want to have a toolbox that is somehow using a graphic user interface. But those toolboxes, they have all some sort of source localization implemented, and they're good. And there's usually a strong community behind it, and it really depends what you're after. But I would suggest checking those toolboxes and then pick the right one that fits you best. And with this, we already arrived at the third question of our poll. That is, why are constraints needed for distributed source imaging? Is it because of aesthetic reasons? Is it because the number of sources are way bigger than the number of channels? Is it because the number of sources are smaller than the number of channels? Is it to practice math? Or is there a non-constraint needed?
Okay, 36% voted so far. Let's give it some more seconds.
Mm-hmm.
And let's share the results. So number of sources more than number of channels. 64% has the highest vote.
Yeah, that's correct.
Yeah.
So that's that. So here we have definitely correct number. It's funny that 3% were picking choice 1, but it's a clear correct answer is answer 2 or B. So now I want to show, after I was introducing source imaging technology or the principle of source imaging, I want to talk about 2 recent studies we did applying source imaging technology to answer some research questions. And here I want to mention the framework of mobile brain body imaging. This all stands on the shoulders of Scott McKay and Klaus Gramann, who wrote these seminal papers already a decade ago. And from back then, all the field is evolving and is rapidly developing, right? So here, usually what we do in mobile brain imaging is tracking the motion of a participant walking freely in space or more or less freely in space while recording high-density EEG. Then I combine these high-density EEG recordings with EEG source imaging. As I described before, we have the mapping. We usually make a head model and field model. We compute the inverse model of this high-density EEG recordings. Then an additional step is needed in mobile brain imaging, namely a sophisticated artifact correction. More to that later. But if you have these artifacts that are produced by these additional movements corrected, even then you have the essential question: how is the brain, or how is are the brain dynamics related to the motion, right? So usually we link this, or we bridge this gap using the motion tracking already in the imaging process. So based on this very simple center out movement tasks here done in monkeys, I did a gait study, or we did a gait study to study movements in humans. So what is common to all these recordings I'm showing here on the top, so this is the LFPs in monkeys, the ECOG in human, the EEG in humans, and the MEG in humans, all performing a central out task. What is already quite visible is this beta suppression here, right? So this beta suppression, so it's a beta power suppression that is indicated in blue. And we also have a high gamma increase indicated in red, right? So we did a gait study in humans, and what we observed while people were walking in this exoskeleton is again very similar to these movement studies done in monkeys and humans, a beta suppression over all the gait cycle, right? What I show here is one gait cycle, more or less the average of all the time warp gait cycles. And what you can see here, very similar in these center out tasks, you can see this beta suppression that is present over the whole gait cycle. One gait cycle here is a right leg movement, a left leg movement, and a right leg movement again. So one whole cycle. All cycles are overlapped and averaged, so their power is averaged here. So that was already quite encouraging because it was so similar to very simple hand movements, right? But here there are leg movements while people are walking. And when we were localizing this, where the beta suppression was biggest, we found out that it's biggest in central sensory motor areas, where very close to where the leg areas, the legs are represented in the sensory system, right? And the second feature we observed is this modulation here. And this modulation was surprisingly present at a different frequency. What I show here in the middle is where the beta suppression is maximum, and here I show where this gamma modulation is maximum. And what was the fascinating thing is that there are two modulation cycles in the gait cycle that are somehow correlated to the walking pattern, right? And what was fascinating us the most is why are these frequencies different, right? Because first we thought maybe the beta rhythm is going up and down, but that doesn't seem to be the case. And I show this here on the top. For beta frequencies, the suppression is biggest, while the modulation for these low gamma frequencies is highest, right? And based on this observation, we moved further because we want to also to investigate this high gamma activity, similar to these studies done before. So in this study, there was more sophisticated artifact reconstruction or correction necessary. So if you don't correct for artifacts, and you look at high gamma activity on the source space, you have this widespread lateral huge activity here indicated in red, right? But then we know the spectral profile of the EMG or electromuscular activity primarily produced by the face or neck muscles. They have a very strong activity that is spread over all frequencies, right? So that's very stereotypical for muscle activity. It is that low frequencies are almost not present, but for higher frequencies, more or less all frequencies are equally highly present during the whole gait cycle. And after we were reconstructing these spectral profiles from in our data, we already could locate this, and we found out this is only this lateralized huge spread, right? And then we could just average or subtract that out. And if you have these original recordings, you reconstruct and correct for these artifactual activities, then a much more beautiful or much nicer source imaging pattern is left over that is much more plausible, right? Because all this strong activity is gone, and still these central leg area activities are present. Plus, there is also quite a clear narrow-banded modulation at around 70 Hertz that was shown for isolated movements before. So that was very nice, and so we could also see and look at higher frequencies even when people were walking around. And this high gamma modulation was not in synchronous to this low gamma, but it was conversely, right? And from this, we were moving on, saying, okay, we observed that this feature, this modulation feature, is different from the suppression, beta suppression feature. So probably those guys are two different networks, right, in the brain. But however, we could not observe that in the gait study because these two localizations were very, very close to each other. So I did a finger tapping study. What I showed here is a trajectory of participants tapping their finger about 9 times, I think. So here there was no movement, then they were start to tap their fingers, right? And what I show here is then just the trajectory of the fingers. And again, we have, even before we move, this beta suppression that is quite strong. But when we can correct for the sustained beta suppression activities, we again see these modulations very similar than in the gait study. And that was quite encouraging. When we look at the beta suppression sources and the modulation suppression and the movement phase related modulation pattern, we already can see that these sources are quite different. So here I show beta suppression or beta increase patterns, where you can see that this beta suppression is maximum in contralateral hand areas because it was a right hand movement. It's represented on the left motor cortex. But if you focus on this modulation feature, you can see a profoundly different network or pattern here. And that's what we were already hypothesizing in the beginning, saying, okay, this modulation feature probably has different sources than the suppression. So they might be functionally different. And just to make an illustration, I have here plotted this beta suppression in blue. And if you make a video out of it, you can see there is some visual information to process, and then there's motor activity. And here, what I want to show with this video is time resolution of source imaging results, because this is only about 1.2 seconds, right? You can see highly temporarily resolved how visual information is transformed into motor information, right? There is really like first visual activity and then motor activity. How do we know that this is correct, or how do we know if these estimates make sense? So we can here argument with plausibility, right? Because this is what we observed here is in the gait study, we have lag movements represented in central areas here and here. So this is the beta suppression, this is the high gamma increase. And again, here is the high gamma increase in finger tapping that is lateralized because it's the hand, not the legs. And the beta suppression very similarly. And we can compare this to a different study who did the same mapping procedure, but for ECOG, meaning intracranial recordings using grids, but also doing mapping, but not non-invasive. So these non-invasive patterns I was observing here are beautifully matching here with these invasive mapping techniques, right? So already here we have some kind of plausibility validation, because we know that the hand areas are represented here in the somatotopic organization of the motor cortex. We know that the hand areas are represented laterally, while the leg areas are here more central, right? So that all makes sense and matches invasive recordings or invasive mapping procedures. Final question is, on what spatial level do you plan to analyze your next EEG study? Again, single channel, topographic, dipole localization, distributed, or don't you intend to do any? So maybe we can switch to the quick poll questions.
All right, so around 60% voted. Let's close the poll and share the results. So it seems that 41% would like to use topographic analysis in their next study, and then 33% distributed sources, 23% dipole localization.
Nice, that's very encouraging. Yeah, that's very interesting to see. So I want to dive more into validation. As I said before, so far, it was just arguing with plausibility, right? But we can do more hard facts validation. One example I want to show here is comparing the localization results with the resected zone in seizure-free epileptic patients after surgery. So those were patients that suffered from epilepsy, they were undergoing surgery, and after surgery they were seizure-free, right? Meaning there was some brain tissue removed where we were assuming that there was the source of their epileptic seizure. And even before the surgery, those authors did source imaging, and then they compare where is the localization of this spike, and is this localization of the spike matching the resection zone, right? And if so, then this is the argument, then the source localization was correct. So you can use different methods and compare to the ground truth surgery location. And this was done in a lot of patients here by Brokeback and colleagues. And what they could show is that the sensitivity is the highest for high density EEG. It's even higher than for MRI, PET, or low density ESI or SPECT, right? That's already quite encouraging. So more or less, the message here that it is for localizing epileptic spikes, it is the best technology. Meaning, if the sensitivity is the localization was in this zone, and there would be seizure-free afterwards, and specificity here means the location was outside the resected zone, and they were not seizure-free afterwards, right? So maybe the surgery was not 100% helpful for those specific patients that happens. And another way how to compare your invasive, your non-invasive recordings with invasive recordings is this comparison to validate, right? So you can do another validation comparing invasive with non-invasive recordings using different inversion methods, and the one that is closest to the real locations. For here we had intracranial and non-invasive recordings, and we can just compare these intracranial localizations of the electrode grid and look where the spike was maximum on which electrode, and if the source localization coming from the non-invasive data is closest to the invasive intracranial EEG, then we know, okay, this is the most accurate or the most correct, right? So here we have a direct comparison because we know the ground truth on the brain, but then we have this non-invasive reconstruction when we compare both of them. And this is a very most recent example discussing a bit more in detail. Is it possible to detect subcortical signals? So that is kind of a very challenging validation study, asking the question, can we detect subcortical signals? Because usually people claim that is not possible at all. And here we had the unique chance to record from 256 scalp EEG channels while having DBS electrodes implanted in the thalamus or the nucleus accumbens, right? So we could record at the same time from these deep structures plus non-invasively on the scalp, right? So what I show here in blue is these electrodes implanted, and what we had at the same time simultaneously these high density recordings in place, right? And what we analyzed is just for about 4 or 5 minutes rest with eyes closed. So when you look at these invasive recordings, you can see the deeper you go into a specific structure here, the nucleus accumbens, the higher or the more pronounced these waveforms get, right? So here these are 3 bipolar derivations from these 4 contacts, and you see the more deeper you go into the structure, the more pronounced these oscillations get. And what is nice here, we have a polarity inversion between this and this, saying probably the electrical source was somewhere around here, right? And if you compare the power spectral density on the scalp and in the intracranial recordings, you can see already with bare eyes that there is a similar peak at around 9 or 10 hertz. And if you then just look at the alpha modulations, the time course of this alpha modulation on the scalp and in the intracranial recordings, you can see very similar time courses. So look at this structure here, or the time courses here and here. You can see also already with your bare free eyes that these time courses are very, very similar. So what we did after these observations is, of course, source localization. So on one hand, we just looked at the non-invasive data. We did source imaging, so non-invasive source modeling, only using the non-invasive data and reconstructing the waveforms for all solution points covering the whole gray matter, right? And then there is a second source of information we got in the study, that is the intracranial recordings. And these intracranial recordings, they provide us as well with the ground truth of this beta modulation, so these beta amplitude fluctuations, right? So what we can do here is we can compare or correlate, in this case, every non-invasively reconstructed time course with the ground truth intracranial waveforms recorded in these deep structures, right? And we know where the true location is given by the implantation site. So we know exactly how the correct waveform would look like and where it would be. And when we do this, what I show here in these hot colors is where the correlation is the highest, and what I'm showing here with these blue dots is where the ground truth location is. Meaning, the closer these red to yellow blobs are to the blue dots here, the better, right? And what we could show here is that the correlation in these deep structures is surprisingly high in areas close to the actual implantation site, but very low and non-significant in all other brain areas, right? Because we did a non-invasive reconstruction of all solution points covering the whole brain, it would be possible that every site would be active, but that's not the case. Only the sites where the close to the true implantation site are significant, and the correlation is quite high. And that was true for all the participants investigated. Based on this, we say, yes, it is indeed possible to detect subcortical signals with non-invasive recordings. And with this already, I want to wrap up. So what we discussed today is how EEG source modeling or EEG source imaging is working. So we discussed field and head modeling. Based on these field and head models, we can combine this information with the actual EEG recordings, using the EEG recordings for topography, combining the head model and the EEG recordings, using specific constraints. We can do or implement the EEG source localization. And in the end, we already talked about validation. So you can compare ground truth electrical signals with non-invasively reconstructed signals. And studies, I was mentioning as example, this research line in mobile brain imaging, where I showed this beta suppression and low gamma modulation having different roles. And we also discussed that it is indeed possible to detect subcortical signals from non-invasive recordings. And with this, I would like to thank my supervisors in the last years, especially Christophe Michel, that it was very influential, of course, teaching me all the source imaging techniques, but also my PhD supervisor, Gernot Müller-Purz, and my project supervisor, Reini Scherer. I would like to thank all my lab and all the scientific collaborators, plus all the funding sources, especially the University of Geneva and the Campus Biotech here in Geneva. With that, I want to thank you for your attention.